Hey guys, I'm Lauren.
And I'm Tyler.
And today is our first day interning at the Salk Institute.
We got to intern at the Salk Institute through the Heithoff-Brody Scholars Program, which is a program that lets us work under a mentor for about 7 weeks, and we get to help them out with their research.
This summer we'll be interning in Dr. Wolfgang Busch's plant biology lab, which is pretty awesome considering that the Salk Institute is ranked #1 for plant biology worldwide. Our mentor is Dr. Wenrong He, but she has us call her Karen. Today is our first day in the lab, and so Karen is going to be showing us around and explaining our summer projects to us. She's going to have us focus on 2 different projects, but we'll be working together a lot, which means we can help each other out. For my project, I'm going to be focusing on chemical influence on root development. So what this means is I'm going to observe whether or not plants can withstand chemical treatment.
I'm going to be using a method called CRISPR to manipulate genes and cut out certain sections of the genome. We're going to see how that affects the physical characteristics of the plant. We're both using a wildflower called Arabidopsis thaliana, and we're going to use those as our test subjects.
It's super exciting to have this internship here because we get to experience what it's like to work in a real lab. There's a little more than 10 other scientists in the lab, which means there's always something else being done, whether it's experiments running or results being analyzed.
We have a lot of prep to do before we actually start our projects. We have to understand the controls and the variables, how to operate different machines that we'll be using. We also have to know how to avoid contamination so our results are actually what they say. Overall, we just need to understand each step to our project and why each step is important and how it relates to the world as a whole. So there's definitely a lot going on in the lab at once, and it can be kind of challenging to keep up sometimes.
I wouldn't say it's overwhelming, especially since Karen is doing a great job at helping us through the whole process, but this whole internship definitely is a challenge for us.
It's really all about asking the right questions, so you have to not just know what you're doing, but you have to know why you're doing it and how it relates to your project as a whole.
Now that we're a couple days into the internship, I'm finally getting a better understanding of what my project actually is. And so for us to test the chemical influence on plants, we have to expose the plants to the chemical. We do this by planting the seeds in a 96 well plate, and each well has a different chemical, except for the first column. The first column is our control. Also, we're going to be using 5 different accessions of Arabidopsis, and each accession is just a slight variation of the plant, but it's still the same. The seeds are going to incubate in the 96 well plate for 7 days. And then afterwards, they're going to grow to be little tiny plants, and then we're going to transfer them onto a vertical plate using a fume hood. I'm actually really nervous to do this step because Karen's really emphasized how important it is to not have contamination, but even just one tiny dot of bacteria can ruin the whole plate. For the next step, I need to learn how to use the imaging system that we have here. Our imaging system is made up of 8 scanners and can hold 16 plates at a time. What it does is scan the plate and then upload the image of the plate onto our server. When we analyze the root length, we can see how the chemical affected it. From there, we can pick which chemical we want to further study. And we'll be doing this by testing that chemical at different concentrations with Arabidopsis.
I've learned so much about my project over the past week. It's actually super interesting. So we're doing a method called CRISPR to knock out a target gene in the Arabidopsis genome, and what that should do is just turn off whatever that gene codes for. We think it codes for longer roots, so if we turn it off, our genetically modified plants should have shorter roots in the end. It's kind of like a reverse experiment of sorts. I just think it's crazy how we can make a personalized tool at the molecular level for my specific gene in my specific project.
Since we only have 7 weeks here at Salk and we need to conduct our research and put together a presentation for the final program, it's kind of nerve-wracking having a time constraint on it. It adds a lot of pressure, but it's a really good thing that there's other interns in the program, and so we can all relate and know that we're in the same boat.
Everyone in the lab is either a graduate or a postdoc, and we just graduated high school, so we have a lot of catching up to do. It's not impossible, but you can't be an expert in just a week or two, so we'll just need some more time to work on it.
Whenever we do anything, we need to record it down in our lab notebook so that we know exactly what we did and we can refer back to it later. For my project, I'll need to collect data on how the plant roots with the chemical treatment grew and compare it to how the plants without the chemical treatment grew. I'll be able to get this data from the pictures that our imaging system uploads onto our server. Sometimes I'm not really sure what I'm doing for my project, but Karen does a really great job of helping me understand.
Working at Salk is so much different than working in a classroom lab. Instead of going for a right or wrong answer, we just want an objective answer. And even though we want our data to support our hypothesis, we have to be objective and record it as it is, even if the experiment was a complete failure. Instead of going for an A+ like in the classroom, now we're just going for an honest answer. And even if our experiment was a failure, that data is still useful to other scientists.
We're finally a couple weeks into the internship, and I've got good news and bad news. The good news is we got paid today, but the bad news is we have some problems. So in our experiments, we try our best, but sometimes it just doesn't work out right. By this, I mean we have contamination. I think the contamination came from when I was transferring the seeds from the 96 well plate onto the vertical plate, and it's really frustrating because I took maybe 2 or 3 hours being so careful to avoid the contamination, but still there's that one dot of bacteria that got on it. It gets really frustrating because it makes the whole plate unusable.
So like Lauren, I have also run into some problems with my experiments. So we put our samples through a gel, which organizes them by size to help us see if we have the right sample. But a lot of times the concentration is too weak to be used, or it just doesn't show up at all, which is really disappointing because if it's too weak, we can do gel recovery, which is another like 3 hours until we actually get our bands back and we can run it again. But if it just doesn't show up, then we have to do DNA extraction again. That's a whole nother day of work that just sets us back. The plasmid that I work with can be difficult, so sometimes it doesn't transfer into our bacterial cells. If it doesn't do that, then the whole plate just dies, and we waited 24 hours for this plate to grow, and there's absolutely nothing to show for it. What I learned is that you really have to just take your time and make sure that everything you do is right. And even if there is no user errors, and there is a lot, trust me, but even if there is no user errors, the experiment can still fail and put you back.
So we finally got some data from comparing the plants, and we can see 3 things at first. 1 is that the plants with chemical treatment are obviously affected. 2 is that plants of the same accession grow similarly, and 3, plants of different accessions grow differently. This means that the chemical treatment is influencing the gene expression within Arabidopsis. So our next steps from there are to see which gene is involved and how it's involved.
A lot of times in science, instead of ending up with an answer, you just end up with 3 more questions. So we have our variable group and our control group, and we were expecting the route to be a little bit shorter in our variable group. It was actually the exact opposite. It was a little bit longer. So instead of just throwing away our experiment and just saying it didn't work, we have to figure out why it didn't work. So our first step is to delete a section of the genome before the gene, called a promoter, and we're thinking that that's going to change the phenotype. And then also we're going to expose the genetically modified plant to different growth conditions and see if it changes if we express different amounts of light or water.
Today is our last day here as part of our internship program at the Salk Institute, and it's a pretty bittersweet feeling. Even though I'm excited to enjoy what's left of my summer, I'm really gonna miss it here. It seems like the last 7 weeks passed by in the blink of an eye, but the experience that we were able to gain is priceless. I loved learning new ways to conduct research, to use more advanced equipment, and to deal with projects that actually have an impact on the world.
Working at Salk over the summer was truly an amazing experience. This is my first ever job, and I'm working at the #1 plant biology research institution in the world. Let me say that again. Not just in San Diego, not the US, but the entire world. The stuff that we're working on is so groundbreaking in the field of plant biology that we're not even allowed to tell you the name of the gene that we're working with. And I find that absolutely amazing. I truly believe that this internship was a once-in-a-lifetime opportunity, and I'm even more excited to learn about biology in college, and maybe we could even come back next summer and continue our research.